now to introduce to you globally respected Dr. Lucienne Ronco. And I have a long bio, so hang tight. Dr. Ronco has been a collaborator and recently a consultant to the FSHD Society. She's a scientific leader with an extensive experience in FSHD translational science and clinical trial design. She has led global teams supporting collaboration and advancement in our understanding of FSHD. She is also an OG of FSHD. Original gangster, that's what that stands for. Lucienne received her undergrad degree from Tufts. She earned a PhD in biological chemistry from UCLA and then returned back to Boston, because that wasn't enough, Harvard Medical School. She worked in both pharma and biotech as she advanced to become vice president and a global scientific leader of basic research and clinical trial design. She's a slacker. So while at Fulcrum Therapeutics, Lucienne participated in the FSHD molecular and imaging characterization as she discovered, designed, and developed methods to assess how well new therapeutics work in human clinical trial designs of FSHD. That was a mouthful, but she did it all. At the Broad Institute, she oversaw the development of rare kidney disease, small molecule therapeutics, and biomarkers. And previously, she worked at AstraZeneca for a decade on oncology drug development. Lucienne today is going to help us understand the process of drug development and the entire clinical trial process. She's also then going to review the current clinical trials for FSHD. And on a personal note, we at the FSHD Society are so extremely privileged to have Lucienne working with us, working with you, our families living with FSHD. So let's hear it, Dr. Ronco.
If I knew she was going to read all of that, I wouldn't have written so much. Thanks, everybody. It's a real pleasure to be here. I started working at the CSO for the Society in December, and I have to say, coming from industry, large pharma to biotech to now working in a patient advocacy group is such a pleasure. And it's a pleasure to be more deeply rooted with folks who are affected by FSHD and to be able to really continue the application of all the knowledge that we collected at Fulcrum and working with all the international folks. So it's a real pleasure. So I am, as Beth said, going to walk you through clinical trials. I have to tell you that decade at AstraZeneca, you spend a decade understanding clinical trials, and working with large pharma gives you a real opportunity to do that. And then you go to, this is a typical course of people going from large pharma where they get all that full knowledge and then they go to biotech and then they can really apply it. So I'm going to work to teach you a bit about clinical trials and a bit about each of the molecules that is coming forward for FSHD. It's complex. And so if you want to catch me, I don't think we're going to have time for questions, but there will be a whole panel with all representatives from all the different companies here, and I'll be moderating that. We've had your questions come to us previously, but also you've had an opportunity to chat with people. I'll be available after too if you still have questions that you'd like to ask. All right. So molecular biology 101. I got a C in genetics in college because I started as an international relations major, and it took me a year to really figure out what was going on. And I know it now, but it's a challenging area to really understand. I think, as everybody knows, DNA makes up the basis of who we are as humans and as individuals. Since within the chromosomes that are wrapped around proteins there on the left-hand side of the slide is the DNA. And it's really an issue with the DNA that drives FSHD, and I think you're all very aware of that. And in healthy cells, and even in with all kinds of genetic diseases, there are epigenetic controlled regulation of gene expression. It says they're epigenetic controlled access to the DNA. And the epigenetic control keeps that DNA very tightly wound up so that it can't be accessed by all these other proteins, the transcriptional machinery that drive the production of RNA and then makes that RNA available for translation to generate proteins. So it goes from DNA to RNA to proteins. That's our dogma. And the dogma is held up, but what we've understood over the past 50 years is there's just so much complex regulation of how transcription occurs in any cell, and all different cells in your body are making different RNAs and therefore making different proteins And those proteins are important for the function of each of one of your cells. Like a kidney cell makes a different protein than a brain cell, than a muscle cell. So that epigenetic controlled access is something you inherit from your parents and your grandparents, and it's usually very stable, and it's passed along. So moving then to what a healthy DUX4 locus looks like. So on the top right, that dogma is still sitting there, but we've kind of made it look like, so a chromosome has a bunch of reference genes on it, and so we have reference books there. And underneath the red line, it is a strict-looking librarian saying, this is epigenetic controlled access, and sorry, we're closed. So when you've got epigenetic control with methylation of a region that blocks gene expression, that's the sorry, we're closed. And that's in the DUX4 FSHD arena. That's what matters here, is we've lost that control. I'll talk about that in the next slide. Yeah. Epigenetic means that it's not that the gene itself is changed, but there are proteins called methylases who can put a tag on a piece of DNA, and that tag causes the DNA to coil tightly. And so it's not genetic, it's epigenetic. It's on top of the DNA, a tag that blocks the DNA from being transcribed. So it basically keeps it so tightly coiled that the transcriptional machinery can't get there. So the recipe, the DNA is effectively the recipe. That sequence defines what a protein will eventually be. The transcription is when the DNA there's a transcriptional machinery, it comes in and it makes just this short little piece of RNA that is then which serves as a specific recipe, as we have there, a specific recipe for a protein. And then the translation is where that RNA is turned into a protein. So in a healthy DUX4 locus, the sorry we're closed is on. Epigenetic regulation keeps the DNA tightly wound. You don't get transcription of DUX4, and you don't get translation of DUX4 because there's no RNA around. And then you don't get the cookie protein. You don't get DUX4 as the protein. So that's a healthy locus with DUX4 shut off. And you'll be seeing as I talk along that we've got new therapeutic approaches coming at, and then the reason we're describing this to you is we've got new therapeutic approaches coming at most every portion of the regulation of DUX4, and we'll talk about that as we go along. So what happens in a FSHD locus? The librarian is not available, and the epigenetic controlled access, those methylations that I talk about that sit on the DNA, that keep the DNA tightly wound, are not there because of that contraction of the D4Z4 domains that I think many of you have heard about. So the open for business, the access to the DNA is there, transcription occurs, we get RNA produced, we get the protein produced, and then I put an image of the DUX4 protein has actually been crystallized, bound to DNA, and those blue coils and the pink coils are actually two coils of DUX4 bound to DNA where it does all of its work. DUX4 is a transcription factor, and how we define transcription factors are proteins that bind to DNA and turn on downstream transcription and translation. So it's complex, and that's why it took scientists so many years to understand what was going on with DUX4. There's more complexity than we're showing here, but this is really what you need to know to understand the therapeutics that we'll be describing. So how many ways are there to treat FSHD? I'll start on the orange, those orange stars. I'll start on the top and then I'll go to the bottom. So we can correct the DNA sequence, and D4Z4 contraction or shortening has occurred in many FSHD loci, so we can correct that. We can block or we can remove the RNA that comes from the gene. We can block or remove the DUX4 protein by degrading it or by other methods. We can repress access to DUX4 DNA, and that's where you actually put those methylation signals back on to the DNA so that the machinery can't, so it gets coiled up again and you can't make the RNA. We can interfere with DUX4 transcription, and we can interfere with DUX4 translation. And all of the basic science that's gone on since we've understood what genes are and how transcriptional regulation has occurred has really contributed to our ability to impact or attack this process in all of these different ways. So I have the DUX4 protein there, and as we know, expression of DUX4 protein typically occurs at the 2 to 4 cell stage in a developing embryo. It's not supposed to be expressed in muscle or any other cell in an adult or in a maturing individual, and its expression leads to muscle cell death. So again, our major approaches are to reduce or eliminate the cause of the muscle cell death, that's DUX4. And once we've accomplished that, our primary goal, we can do additional things. We can help muscles to grow or regenerate because we've had muscle loss as a result of DUX4 expression. And fibrosis and fatty infiltration are important mediators of the loss of muscle function or downstream results of the loss of muscle in a muscle fiber. And there are small molecules out there that are being tested to see if they actually could reduce fibrosis. So the regeneration and fibrosis as a secondary approach, once we've reduced DUX4, are, I think, very exciting opportunities for the community that are being really actively engaged now. So this is that thing that I told you took me 10 years to really fully understand. It's the overview of therapeutic development. And I talked about basic research, and there on the left is a scientist peering into a microscope. There's a lot of preclinical work that must happen before you can even think about taking a molecule into the clinic. You've got to understand, why is DUX4 on? And why is it only expressed in some individuals and not in other individuals? There's complex regulation that we didn't even discuss yet, but those are the disease mechanisms. Drug discovery can be a very long process. It can be, we've learned so much that we're a lot faster with it these days, and making gene editing approaches can be a lot faster as well, but it's still a long process, and we have to use animal models to really understand disease progress. And the thing that may not be apparent to you is that we make some models in mice, but mice aren't really humans, and a lot of their systems don't recapitulate our humans and our human systems. And currently a mini pig has been made with a DUX4 locus introduced, and they're larger animals. They have more similar systems to humans. And so what I mean to say by saying this is that, science never sleeps. It's like rust never sleeps. We're continuing to build new models, new approaches, better models. And I think a lot of the new therapies that are being developed will be tested in the pig. And that allows us to start to understand, so if we reduce DUX4, do we reduce it all throughout all muscles? If we only reduce it in some muscles, what happens? What happens to the fat? What happens to the fibrosis? What happens to the inflammation? These are all very important questions that we don't fully understand. We keep building better preclinical models to better answer those questions. The clinical research is one of the most important things that I think happens. It's those natural history studies. And I can't encourage you enough to participate if you haven't already. I'll be talking a little more in detail about them at the end of the talk, but it's the way that we really understand the course of the disease. And it's only by that understanding that we know what to measure in a clinical trial. We did a lot of work in those 5 years at Fulcrum, and the following therapeutic developers are continuing to do a lot of work, as well as the natural history scientists, to understand what physical assessments, what biomarkers we should be using, and how to design our trials. So the clinical trials, we have to do a lot of work. If you haven't taken a molecule like losmapimod that already been in humans for other diseases, we had a good understanding of what level to dose at, what was a safe level, what was an effective level. If you haven't done that previously, you've got to do that in your phase 1. What is a safe and effective dose in humans? You look for the safety and the side effects, and you look for efficacy against the disease. And typically in a phase 1 study, you're looking at a very immediate biomarker like DUX4 regulation or DUX4 gene regulation. And then a lot happens between the clinical trials and the launch. It can be a year, it can be 2 years, it can be quite a period of time. Once you've started a phase 1, do a phase 2, oftentimes you might expand a cohort or go to a phase 3 prior to even thinking about doing a launch, where you look for regulatory approval, You have to educate the regulators about FSHD, and we've spent a lot of time doing that. We as a community have spent a lot of time educating the regulators. What is FSHD? How does it impact people over the course of their lives? How abling or disabling is this disease? And we have to also educate the payers to show the impact to individuals through the course of their life so that they make decisions that they're going to take on the paying for these new medicines. And then there are longer-term safety studies that can go for 15 years. Sometimes there's a requirement to evaluate that therapeutic for up to that period of time to understand. I was telling folks yesterday, sometimes you have to take a molecule into thousands of individuals before you can truly understand the safety implications. Think about the COVID vaccine. Went into millions of people so rapidly. We knew the safety of that very rapidly. But for something like a rare disease, it takes longer, and it takes typically thousands of individuals to understand the safety. So we don't have to spend too much time on this slide. It's just talking about phase 1, 2, and 3. These are flexible. Companies can take a phase 1, 2 approach in rare diseases so that you go right from a phase 1 to a phase 1, phase 2, and within 3 to 18 months, you could have answers regarding a novel therapeutic. The number of individuals, that's pretty typical, that you've got tens in a phase 1, and in rare disease, you could have 50, 60 in a phase 2, and that's adequate to demonstrate oftentimes your biomarker modulation. And then the phase 3 can be hundreds to thousands, as I said. I won't talk much about the safety purpose, but the percentage of drugs that move to the next phase, I think the 25% is generous. So the 70% in phase 1, then 33% in phase 2, that's pretty reasonable. But I think the numbers that go on to phase 3, they only go on to phase 3 if they've shown some efficacy in some of the endpoints in the earlier studies. And that's a very challenging bar to hit. Very challenging. I think that especially with small molecule development, but I think with gene editing, those numbers are higher, and maybe the 25% is accurate. My husband is a basic scientist, and when I'd get discouraged at times, as I've been involved in drug development, he's like, "Why are you discouraged? You know it's only going to work 2 to 5% of the time." And I'm like, "Yeah, but we've worked hard to get here." All right, so I'm going to now step through a number of studies, and mostly only those studies that are in humans already or will be in humans by the end of this year. Companies that have projected that they'll be starting later than that, we're not going to have a detailed discussion of, but we'll have representatives from those companies here, and we'll be asking them a series of questions. So losmapimod was developed by Fulcrum. It interferes with the transcription of DUX4. So it blocks the creation of the RNA from the DUX4 gene. So it can't make the protein can't go on and activate its targets, and it doesn't cause the muscle injury. That's what the preclinical data very clearly showed. It was a repurposed drug, and because it had been in 3,600 previous study participants, we knew the safety profile. The REACH study is set to actually complete by the end of this year, and I don't expect data this year, but perhaps by the beginning of next year. The society anticipates there will be an open label extension. That's what Fulcrum has said. And I won't go into this too much as it's a fully completed study. And again, it's completed. It was in US, Denmark, France, Germany, Italy. And again, we're expecting that data quite shortly. We'll speak now to the Roche studies, manoeuvre. They've produced a molecule, GYM-329. It's an anti-latent myostatin antibody. What does that mean? So myostatin is a negative regulator of muscle growth. So we are very complex. It's kind of shocking. We get up every morning and can function. I'm often astounded because you learn so much about science and you realize how complex we are. Myostatin blocks muscle development. So if we can block the block, we can allow muscle development. And so in this case, Roche has developed an antibody against myostatin that should and does, in preclinical models and in humans, been demonstrated to enhance muscle growth and it binds to the inactive myostatin and blocks its conversion to an active version of it. And as I said, the preclinical data demonstrates increase in muscle mass and in strength. So the duration of this is a placebo-controlled study, meaning that there will be individuals, half of the individuals will not receive the medicine. They'll get a placebo, half will receive this antibody. It is going for 52 weeks, so a year. There are wearable devices, which is a very exciting new advent in the therapeutic trial, and MRIs as well. It is complete. And I don't see here immediately when it will read out, but we could ask our Roche representative, Angela is going to be here to tell us when we might be hearing about the readout of the manoeuvre study. Roche also has a reinforce study that's being run by Sabrina Sacconi, and any of you who might have been here previously, she helped chair our scientific meeting that we had. She's in Nice, France, and she has identified that a molecule that's typically believed to be an inflammatory mediator, interleukin-6 or IL-6, is upregulated in the blood of FSHD subjects. And we can take molecules that have been developed for other disease purposes, like an antibody against interleukin-6, and apply it in FSHD. And this is what she's doing. So the antibody is called satralizumab, and they have horrible names, sorry. But it's always good when they're shortened and made easier to understand. And her hypothesis is that inhibition of IL-6 by having an antibody that binds to the IL-6 receptor in this case would reduce the activity of IL-6, and that should have an impact on FSHD, and that's the hypothesis that she is testing in France, Ottawa, and Ottawa, Canada as well. All right, so now I'm going to move on to the Avidity Fortitude study, and I think it's made a lot of news this past week where Avidity released data on Wednesday morning that was quite exciting, I think, for the community. So Fortitude is a complex medicine that is a monoclonal antibody against a transferrin receptor, and the transferrin receptor is on our muscle cells, muscle fibers. They are using that antibody to direct their reagent to the muscle, and their reagent is an siRNA that binds to and forces the degradation of the DUX4 RNA, thereby reducing the ability to make the DUX4 protein. It was called AOC 1020. Yep. And this is complete, of course, and they've reported it just this past week. Is there anything there we want to speak to? They did the biopsies in after having an MRI in leg muscles of subjects, and it was a small and focused study of 12 individuals for their first evaluation, with 4 receiving placebo and 8 receiving medicine. There is an opportunity to get involved, and once I show you the data, you'll understand why I wrote there in that little blue box on the top right that an expanded cohort is enrolling now, and that's because of the preliminary data from the very lowest dose of this molecule 1020. So its new and abbreviated name is Delbrax, and so that's what we'll call it going forward. This data was just reported Wednesday, and I pulled the data from their presentation that they made on Wednesday morning. Also, Jeff Statland, whom I think many of you may know, he's an innovator and leader in the clinical trial network for FSHD. He presented this work for the very first time to the scientific community on Friday. So these are some of the pieces of very exciting data that came from his presentation. And I draw your attention to the very middle one with the yellow bar that is going down with a blue line that shows a reduction of 53%. What Avidity has measured there is a set or a panel of transcripts that are upregulated by DUX4 expression. We can't measure DUX4 directly in clinical trials very easily. It's very challenging to find, but we can find the transcripts that are modulated or upregulated, driven to be expressed by DUX4. And what they've shown, PBO is placebo, and the right-hand side is Delbrex with that red block going down. A 2 milligram dose, 2 milligrams per kilogram was their lowest dose. They show a 50% reduction, greater than 50% reduction, meaning they have now only found half of those DUX4 gene transcript levels to be expressed in the second biopsy that they took at the end of the study. That's very exciting. That was the goal of their trial, and that's a great outcome. On the bottom left, I have Delbrex improving muscle strength. It's obviously the next important question. If we reduce DUX4, do we, over the course of the study, improve muscle strength? They have evaluated a number of endpoints in both upper body and lower body, and I know you're all aware of all those different endpoints. And I'll just point to the shoulder abductor, where that orange color moving to the right is showing improvement in the strength of the shoulder abductor compared to the gray, which is the placebo controlled set of 4 subjects. There's a bit of an overlap there, but there is improvement in a movement to the right. And then in the ankle dorsiflexion, you can see as the gray moves to the left over the course of the study, there was decline in the strength of the ankle dorsiflector. With the treatment of Delbrex, there was improvement. It's a small study. It's not a long study, but it's showing that the trends are moving in the right direction for muscle strength. Then another very important outcome that therapeutic developers are interested in and the regulators are very interested in the United States and in Europe are patient-reported outcomes. That means that the patients or their doctors, and I didn't show the doctors, I actually only pulled the patient-reported outcome here, report that they feel better by a whole variety of questions. And there are a number of patient-reported outcomes that were used in this study. Again, in the orange, with the treatment with Delbrex, it's moving in the trend that you would anticipate if you were seeing improvement of the disease. And the gray is what's shown with the placebo treatment. So many indicators from this study are moving in the right direction, which has given Avidity a reason to continue the 4 milligram per kilogram study and then go to an expanded cohort where they'll choose the dose that they intend to use there. Okay, so what's coming next? There's a lot of approaches, potential approaches, as I showed you in some of the early slides. Arrowhead is developing ARO-DUX4. They have a platform, the TRIM platform, which is short for targeted RNAi molecules. They've got unique sequences that should have been demonstrated, should I say, in preclinical studies to target DUX4. One of the main things about the RNAi approaches is that we use our knowledge to stabilize these molecules so that they persist in the muscle for a long time, and that's what stabilizing chemistry means. Linkers, you've got to have the right linkers, and I won't go into that. But they also have targeting ligands, just like the transferrin receptors I was describing for the Avidity molecule. Arrowhead has their own targeting ligand to get the molecule to muscle cells. So here's the study design. Part 1 is just a single dose, which would be done for safety considerations. And then part 2, there will be doses every 3 to 6 months. They intend to go for 360 days. They too will do a 1-to-1 dose. They expect 20 visits. There will be MRI. There will be leg biopsies. It's beginning early in 2024, and it will occur in New Zealand and in Canada. And you can see that it's the typical inclusion-exclusion criteria, 18 to 70, FSHD1, with a clinical severity score of 3 to 8. So because this is actively enrolling, I wanted to go through that component a bit more, as opposed to the previous ones that were complete. So anybody intending to go and live down in New Zealand or in Canada, or already living there, have an opportunity to take part in ARO-DUX4. So coming soon, and they intend to begin to dose their EPIC molecule, EPI-321, this year as well. So this is a different approach, and it goes back to the epigenetic regulation of the gene that I was describing earlier. EPIC's intention is to re-methylate the D4Z4 region. So in the top left of that blue box, you see a stretch of blue with a bunch of blue dots along the top leading to the DUX4 gene. That's a DNA sequence, and those little blue dots are methylation moieties that are attached to the gene, to the sequence of DNA, and they keep DUX4 turned off. In FSHD, and if you move down to the next one with those open circles, there is something we call hypomethylation, or low levels of methylation. So the D4Z4 regions that remain upstream of the DUX4 gene are not methylated to the extent that they are in healthy muscle cells. It is de-repressed, which means it's turned on, and we get sporadic expression of DUX4. The EPI-321 approach is to tether a methylase to that DNA piece and have that active enzyme, it's an active protein, replace the methylations onto the DNA, thereby shutting off DUX4. It's a very interesting and very exciting approach, and it would reduce expression of DUX4. They also expect this could be long-lasting because, as I told you, methylation is usually stable through generations. So if they're able to repress it by applying that methylation sequence, a series of methylations to that sequence of DNA, it could be a long-lasting effect. And it uses the CRISPR-based technology. And you could ask more about that approach of Wes when he's here in the panel discussing EPI-321. So again, it's intended to begin later this year. Let's talk about who can take part, the inclusion and exclusion criteria, 18 to 75, FSHD 1, a Ricci score of 2 to 4, must be able to walk 10 meters because they'll be using probably the 10-meter walk run to be able to evaluate function, MRI, muscle biopsy. All patients will receive the drug, so in this case, no placebo. They expect to start with 6 to 9 subjects. And they intend to do a 4-year follow-up because, as I said, they anticipate it could be long-lived, so a 4-year follow-up is appropriate to understand if indeed they do get the long-lived effect that they hope to see. It will be run in the US, UK, Canada, Germany, and the Netherlands, and so you have to stay tuned for more information from Epic Bio as we wait to see when they might initiate that study. And there are many more therapies coming down the line. As I said, we only made slides to those that were beginning this year. But, you know, we got to watch this space. The Society will continue to keep you informed from our website. We're always willing to engage questions and help direct you to the right people as well for other questions that you may have. Now I'm going to talk a little bit about the natural history studies. I told you previously that they're exceptionally important. And it's also, they're still ongoing, even while we have these therapeutic approaches happening, and we do need them to continue to enroll. And you can see Jeff Statland, who made the presentation, is there evaluating, I think, ankle dorsiflexion in that case. So why are they important? We're still understanding what are the best outcome assessments and measurements of disease that we should use in our clinical studies. It helps us design better studies, because if we have the wrong outcome assessments, a good drug can fail. And so these allow us to understand what are the best assessments, and it helps clinicians provide better care. We've had a very interesting and exciting presentation earlier this week where MRI has been done for the whole body for a number of these studies, but there's now an academic lab that's doing whole body MRI. And what they showed us is that it's not just a handful of muscles, and you guys well know this, but this is great information to take to the regulators and to the payers. It's not just the trapezius, it's not just the deltoids, it's not just the tibialis anterior, or the muscle that allows you to pick up your foot. They found fatty infiltration in almost all of the 118 muscles that they measured in a small group of subjects, but it's very exciting. No, it's very valuable data for us to use to talk to the regulators to say, most muscles are affected by FSHD, and patients know this, but now we can demonstrate it with data to the regulators and payers, the real impact of the disease. And it's that bottom line, helping clinicians and scientists better care for people with FSHD, better understand FSHD, and better inform and educate the regulators and the payers, which is now going to be a very important component of what the society is going to undertake. So what happens if you get involved in a natural history study? At least 3 study visits over a number of years. You perform strength and movement tests, you fill out questionnaires, and for Move Plus, there are blood samples, saliva samples, MRI, and muscle biopsies. The reason we ask for the MRI and the muscle biopsies is it allows us to say, by MRI, how much fatty infiltration is there in those muscles, and can we find DUX4 there? And what else can we find there? And so therefore, what else can we target therapeutically going forward? So it becomes very valuable information as we seek to continue to strive to reduce DUX4 and then to accentuate or activate regeneration and a reduction of fibrosis. And that's why we continue to ask for the muscle biopsies. I know it's a burden. I know it's painful. There's really good reasons to continue to do that work. Who can join Move and Move Plus? Anyone with a confirmed doses of FSHD 1 or 2. You have to be between 1875, have lower leg weakness, be able to walk 30 meters without assistance from another person. And those blue dots are showing that Move Plus, Jeff has been so successful in expanding his clinical trial research network across the US and Canada. And you can contact Michaela, you can contact the society if you're interested in being involved. Okay, so you can get involved now also in clinical trials. The Avidity clinical trial, we've understood from them, they're going to expand a cohort and continue on to do those studies. I believe that's enrolling now. You could be involved in Move and Move Plus natural history studies. So there's other ways to really be ready as an individual and as a family with FSHD. Be connected to the society. We strive to provide information that can be really valuable to you. Please participate in research surveys. Get to know your local neuromuscular clinic. We have understood that, in the absence of a real therapy, patients don't go and see their doctors that often. They don't strive to find a neuromuscular clinic that could really help them be involved in a clinical trial. Perhaps now is the time to reconsider that decision. Perhaps now is the time to find those clinics, find those doctors, build a trusting relationship, and have an opportunity to really be there when the therapeutic successes start to really come through. Get the best care available and stay as healthy as possible. And I think we've strived over, that's obvious, but we've strived over the past couple of days to make all of that a lot of new information available to you. And get genetic testing. Genetic testing is essential to participate in clinical trials. It'll also be essential to be able to receive any novel therapeutics that come along. The society is going to be taking a new effort to get up to 5,000 individuals in the United States and Canada tested, so that when the companies come to us and say, we need more patients, we need more clinical trial centers, that data is there, we know who the individuals are, you've got a CLIA-approved test, and you're ready to go. Okay, and that's it. And I don't think that we're taking questions now, because we're going to have, after this next talk, we'll have a group of representatives from the companies, and there'll be a lot of questions there. But I'm very willing, if there's a burning question, I'm happy to answer it, but then I'm very willing to answer other questions. All right, move on.